Thank you, and thank you for the organizers, right, Maribel and Gabriella, and of course Hector as well, for welcoming me and everyone here to this wonderful meeting. I really been enjoying it, and the really interesting talks that we've had today. So, and before. So, most of what I'm going to talk about today is as an approach to addressing problems that are perhaps difficult to think about experimentally. In a way, it's inspired by Cuatro Ciénegas, since Cuatro Ciénegas makes us think about big problems and ways of looking at problems that we might have not other imagined, because the kind of life that's at Cuatro Ciénegas is unusual, and we have to think, so how is this possible here? And so it's really inspired in many ways by the kind of work that's gone on here at Cuatro Ciénegas, and I've been so happy to be a small player in the work here. So if one thinks about diversity and patterns of biological complexity, one can look at Chiapas and say, well, how do we have so much diversity of life, and why do we see the kinds of organisms that we see? In Minnesota, where I'm at, there's also diversity, and we wonder, well, why do we see the many different kinds of flowers that exist? One can go to the other side of the world, in Japan, and see also quite a lot of diversity that's not as easy to see on the screen, but it's there. And one can, in fact, even go back in time, slightly, dated photo of the Cretaceous, and it's there's a lot of diversity there. Here we finally have something that's animal-like, and the big questions in ecology and evolution have been: why do we see the diversity that we see? How is it maintained? Is it stable? Is it unstable? What stabilizes it? Those are all the big questions that we've had for well over 100 years now. And a lot of the approaches that we've taken have evolved. Many of the talks that we've heard today. So it might involve genes, evolution, environment, all sorts of different aspects to it. And in the short term, these are quite often helpful. But to think about the sort of expanse and sort of including the diversity of life at Cuatro Ciénegas, is this enough to understand the kinds of things that we see when one looks over both bigger scales and perhaps longer times? And so what I'm going to talk about today is sort of an approach to understand this very complex life that we see, partly by making it a lot more simple. And maybe that's not fair, but that's what we're going to do. And so if we go to back where I work in Minnesota, not necessarily in this field, what you see is a microbial and macrobial world, right? So the world that we see now is very complicated with all sorts of many different kinds of organisms. And what I'd like to encourage you is to remember that, in fact, most of the history of life doesn't have any of those big organisms. And that even though 700 million years or so, when multicellular organisms arose, was a long time ago, most of the history of life is in fact dominated by microbes. So, and almost the entire, if you came in a spaceship and you came by Earth, for most of the history of life on Earth, it would be covered with slime. It'd be a world of slime, which may or may not excite you, but that's what you would see. And so when we think about the nature of the world that we see now, we often forget this long history. And that's what we're going to not forget now. And so the difference between the macrobial world of today and the microbial world of now is the evolution of multicellularity. So there it is in big letters, multicellularity. And so the question I'm going to first ask is, hopefully I'm going to ask, how did this big multicellular, how did this thing evolve? How did we get multicellularity? Recognizing that for most of the history of life on Earth, we didn't have any multicellularity. Okay, so historically the answer, I'm not too loud, historically the answer was very similar to, we were reminded of it, sort of pre-reminded, by a talk by Lakshmi, who mentioned cancer. So for quite a lot of the history of biology, people said that the evolution of multicellularity is a hard problem because of cheating and genetic conflict. That any multicellular organism has the potential to have cancer, and so you always have this collapse. And for sure, cancer is a big problem. And so this was the reason why multicellularity took so long to evolve is because of things like cancer. So most of the time when people investigate this, they've historically done sort of comparative methods, which are very powerful and have told us a lot about how multicellularity has evolved. But it makes it very difficult to actually see the individual steps that occur. And so taking a page from this book by Stephen Jay Gould, Wonderful Life, he suggested that we should think of, he thought of a sort of thought experiment. What if we could replay the history of life and then see how it occurred? Could we learn something new? Would it be different than looking backward? If we could look forward, would you get different answers than looking backward? And so what we do, when we're not trying to, I was looking too far forward there, is we take our microorganisms and we can evolve them in the lab. And so we can experiment. So for Gould, it was a thought experiment. For us who love bacteria and microbes, as we heard earlier today, we can actually do the thought experiment for real by evolving them in the lab. So our experiment that we've been doing now for over a decade is to take Saccharomyces cerevisiae, which is basically yeast, which everybody in this room has consumed and is a friend of, and we grow it in the lab under very simple conditions. After 24 hours, we take a small aliquot out, we put it in a microfuge tube, we let it sit, we might centrifuge it, or we might let it sit on the bench. We get rid of all the extra stuff that the microbes were in, and we transfer it to a tube. So this is the very high-tech approach that we took. And the thoughts are, and this actually follows from the talk we heard yesterday, is that it turns out the bigger you are, generally in liquids, the faster you settle. And so things that are big settle faster than things that are smaller, all things equal. So this is a selection for being big. And turns out multicellular organisms are bigger than unicellular organisms. So after we had 10 replicates and we ran it for 60 days. The ancestor is there on the top, so it looks like a yeast. And here's our 10 populations, and they all evolved multicellularity. It was a very easy experiment to do, and it was very repeatable, and showed how easily it is to evolve multicellularity. And anybody can do this experiment. You can do it at home. It's a little tricky to have sterile media, but it turns out yeast is very common, and you can do this experiment at home. You can do it in a lab. You can do it in a high school, like the Civetta, anywhere. So we call this snowflake multicellularity because in Minnesota we have lots of snowflakes, not the whole year, and they kind of look like snowflakes, at least a little. All right, so because it's yeast, and yeast are one of the biggest model systems for genetics, you can do the genetics, and so we can look at individual genetic changes. So here we found one gene, NBA, who cares what the name of it is, and if you put the NBA gene in the ancestor, it makes it bigger. So the only difference between these two is this knockout in the NBA gene, and this is log size, so it's quite a bit bigger. So just one genetic change gives you multicellularity for yeast, a kind of multicellularity. And it turns out there's lots of different genes that'll do this. So here's a whole bunch of them. So NBA, TUS, AIM, this is actually a pair of different genes as well as this, and here's ACE2. And they give you different patterns of multicellularity. So there's many different ways of evolving this thing, and it gives you different kinds of multicellularity. They all have this sort of general snowflake form, but you can get a single genetic change, and you can have variation in that. So it's really easy to do, and there's lots of ways to do it. And it turns out, if you look at the transcriptomes, so here's the transcriptomes of all of those guys, there's 5 genes which the expression is changed, but there's a tremendous number, up to thousands, that differ. So you see that the similarities here give you the snowflake, but all those other things that are different give you variation. And so it's quite easy to see how variation can arise in a simple system. So remember, 60 days, we all got snowflakes, they all look similar, and yet they all look kind of different. And you can see these differences just in the expression patterns, with those 5 genes that are the expression of which are similar. And those genes, because it's yeast, we can actually know what they do. They're involved in daughter cell replication. So the way multicellularity evolves in this system is that the daughter cells stay attached to the mother cells, and the granddaughter cells stay attached to the mother cells, which whatever. So you just end up with this bunch of cells staying connected to each other, because these 5 changes are all involved in how cells separate from each other. And there's lots of different ways of breaking that system, because there's lots of genes that are involved in cell replication. Okay, so once you replay the tape of life, you kind of think, oh, maybe I can learn more. So about replaying the tape of life. So in this system, so what we've done is we see all these genetic changes that happen, and we ask, once you get a genetic change, does that change the outcome if you redid it? So now we're replaying the tape of life in an alternate reality over again. So we had 10 replicates initially. We took 5 of them, 5 genotypes, and we evolved unis from them. So we back evolved them from multicellularity to unicellularity. I'm not going to talk much about that. That was the work of Maria Rovieta Gomez, who got her undergraduate degree at UNAM, who's now at UC Irvine. And we took those unis and restarted our experiment. We meaning Jolene, the grad student who worked on the project. And she asked, can I evolve multis from those unis which are already evolved from multis? So these are the kinds of experiments you can do with experimental evolution. And this is what she got. So without going into any detail, what you can see is that on the y-axis, we have days to multicellularity. Some of them took a long time. Some of them took a lot less. So this is about 25 days. This is closer to 60 or 70. So depending on who your ancestor was, it took a lot longer or a lot less time. And so not so long, a lot of time, and this is actually the ancestor. We made it a little harder to evolve multicellularity to see if it would matter. So this is not exactly the same selection, but it's pretty close. And the thing that I want you to see, this pointer is really not for me. These are the slow guys. These are the fast ones. They evolve multicellularity quickly. These ones evolve multicellularity slow. All the slow ones have this ancestor. All the fast ones have this ancestor. Let's see if I can do this. So this ancestor is the one that mattered. So this is like experimental phylogenetics, right? And it's the genetic change to the first multicellular phenotype that makes all the difference. So even after only, I don't know, it must have been about 100 days of selection, you can get historical differences that matter. 100 days. Not like 100 years or 100 million years. Even really short amounts of historical differences can have important consequences, at least over this scale, for how big things, literally, how bigger things evolve. So when we saw this, we asked, well, what if we made a bigger difference? So we done everything with bread yeast here, bread and alcohol yeast, beer yeast. What if we took a really different kind of yeast, Cluveromyces, which is dairy yeast? Right, so Cluveromyces is a haploid. It has completely different chromosome structure. It only has 4,800 genes, a lot less than Saccharomyces. So they're really different. It's 100 million years separated. It's a very different organism. And we did the same experiment again. We meaning Will Driscoll and Pooh, who's sitting up there. This is what we got. So if I hadn't labeled them, I doubt, because I can't tell the difference. So that's the Saccharomyces, that's the bread yeast, and here's the leche yeast, right? Not so different, they look identical, even though there's all these differences. And if you sequence them, you find out that in fact, 2 of the genes that are important here are actually all of the genes that are important there. So 100 million years separated, in fact, they give you exactly the same form. So this is a really strong case of convergence. Convergence in a phenotype for organisms that really have quite different habitats, and they live in different places. So these are the kinds of experiments that we are excited about doing, about sort of alternate reality experimental evolution things. The things you can test by looking forward, and not only, so here let's see if I can do this. We take advantage of the ability that you can do this history of looking backwards and ask, does this history matter? And when does it matter? And how does it matter? So you can see that it matters genetically, at least over the experiments that we've done, at least, but phenotypically less so. Okay, so how can multicellularity evolve? Well, it can evolve really easily, at least in the lab, which when different genes giving slightly different phenotypes, and then you have this question, this hypothesis, that cancer risk is really a limitation for multicellularity and that cheating and genetic conflicts, and it's totally wrong. It's just wrong. I mean, it doesn't mean that multicellularity doesn't mean that cancer isn't important. It means that the origin of multicellularity is not limited, at least in this system and lots of other systems, for how easy it is to arise. So whenever we show this, the next question is, why? Why did it take so long? So that's it. But now you can ask that question, right? That's the power. It's like, oh, I can answer one question, let's ask the next. So, well, one, I'm not going to answer it for you because I don't know. I don't know the answer. It turns out you have to work. But one can ask for the critical earlier transitions. So, an important earlier transition is eukaryogenesis. So this, I'm not going to answer eukaryogenesis today, I'm sorry. Again, but we are going to do, so eukaryogenesis is archaea and bacteria come together and they're very happy together and they make a whole nucleate of eukaryotes that include us. And there's a bazillion different theories about why. And there are just so many. And, not worth, they're interesting, and then one of them might even be right. But it's hard to know which one's going to be right. So for us, the thing to do, and then I'm going to talk about a part of an experiment, is to do the things like, well, if we think eukaryotes are a consequence of bacteria and archaea, well, let's grow bacteria, which are, here's a bacteria, and here's an archaea. So if you came to Anna's talk yesterday, she talked about these 2 different ones. These are 2 microbes that are perfectly happy growing in halophytic environment. They're halophiles. They like growing when there's a lot of salt. And so we just propagate them in the lab for a while, 8 replicates, and we did it under 2 different environmental conditions. So rich environment where there's lots of yeast extract and tryptone and a fair amount of salt, in a poor environment where there's not quite as much salt and no tryptone and not hardly any yeast extract. And the reason we're doing this is a preliminary part of the experiment. We want to understand the niche boundaries of these things. And so again, if you were attending Anna's talk, she was talking about sort of the nature of the extremophiles. And so we're doing an experiment that many people have done with mesophiles, with organisms that live in normal, our kind of environment, and ask, what's the boundaries? Why can't they become better at living in extreme environments? And we're sort of asking, what's the niche boundary of organisms that are difficult to grow, and asking how does the niche boundary evolve? So this is a kind of precursor experiment to understanding how these organisms might get together. All right, so after 10 transfers, we already see rapid evolution. This is really, really, really fast. We weren't going to actually talk, so when we were planning this experiment, we figured we wouldn't see anything by 10 transfers. But it turns out we were wrong, that in fact they already evolved. So in in one environment here, in one bug. In the poor environment, the ones selected in rich media don't do quite as well as the ancestor, and they do just about the same, whereas the one grown in the rich environment, when grown in the poor media, is actually, strangely enough, so in the poor environment it doesn't do as well, but in this environment it's doing a little better than the one selected in rich. And hopefully I won't keep rehearsing that, like I just did. So anyway, so this is an example of a specialist adaptation when you're good in one environment makes you less good in the other. In this case, it doesn't matter which environment you grew in, both of you are better than the ancestor. So in this one we see big differences. In this one we see a very big difference, asymmetric difference, but it's clearly a specialist. Which is the archaea and which is the bacteria? I mean, who knows? I mean, it's important to know, but half the time I can't remember myself. Right? So I'm not sure this is anything general to archaea and bacteria. What it was is that you see the bacteria gives you the specialist adaptation, and the archaea gives you the generalist. And so whether this is general to all archaea and halophiles, this is the kind of experiments that no one has done with archaea before, and these kinds of halophiles, and ask how do niche boundaries evolve in general? And these are the kinds of experiments that you're going to need, that we're going to want to do to understand, what's the limits of life, or what's the limits of life for these systems, and hopefully that will give us a better idea about how these bugs came together. So, are there other trade-offs? So everybody talks about, so these are, remember, 10 passages. So it's roughly 50 generations, a little bit more, not a lot. So if you compare, there's a lot, we compared lots of aspects of how these things grew and a whole bunch of different aspects of their environment. And we saw for the, and I did it again, pushed the wrong button, I'm still pushing the wrong button. There we go, okay, stop pushing the wrong button. So in poor environments, and we have one that's poor plus minus some salts, we see that the poor media ones actually don't do as well when you get rid of the salts, the other salts, not sodium chloride. So for the bacteria, we see trade-offs in the poor environment with and without the other salts. We don't see that at all for the archaea, right? Trade-offs occur in rich environments. So when you put less salt in, actually you really get, depending on which, the selected in the poor environment does less well when you don't have as much salt when you're in the rich environment. So it's very complicated. But the argument is, so if you take halophiles and you ask, what are the common trade-offs? The answer is, there are none. There's no trade-offs here, right? There are no common trade-offs. Of course, these are completely unrelated. They're as unrelated as cellular organisms can be. But when someone tells you what are the common trade-offs, well, there might be some, but in the middle of the niche boundary, you don't see them. And this is what we see here, right? So this is the first kind of experiments. We're sort of testing to figure out how we're going to grow these bugs and examine if we're going to see if they can evolve together, how is it that we can ask if they do or not. Okay, so what are the other critical earlier transitions? Let's see how we're doing on time. Okay, so another big one was the origin of life. And the challenge for the origin of life in doing experimental evolution is, well, it's not alive, so how can you study it? That's a little difficult. Well, when you think about the origin of life, people have focused on two aspects, cells and the molecules within them, so particularly replicating molecules. And I don't know how to do that experiment, at least cells and replicating molecules together. So we just focused on, at least initially, on replicating molecules. Can we see something like a lifelike process in a system that's not fully alive? That's what we're asking. So once again, we're going to do this thing where we say we're going to evolve forward and not look back, because now we're at a place before the last common ancestor. So comparative evolution is very difficult, and people are doing it, but it's extremely hard what information you can get. So we do it experimentally. So we have a single-stranded DNA library that we throw on a target. We then wash off the things that don't attach to the target, get rid of the target, and PCR amplify everything that's left. So are we going to see lifelike processes here? Well, the answer is, of course, yes, because it's PCR, and of course you're going to get things. This is non-immunogenic PCR, right? So we're just trying to see how we can get things to evolve. And so DNA in this system has a phenotype, which is unusual to think about. And so the phenotype is, can it stick to this target? That's our phenotype. And because it's DNA, it has a genotype. So we have a genotype-phenotype map that's the same thing. Right? So the complexity of the system, so we've simplified it to something that we can actually investigate. All right, and so we attach it to DNA, attach it to a target, take it off the target, and then repeat. And so we repeated it 8 times, and here single-cell genotypes attached to beads. And here's, make sure which button is which. Here's the number of counts. So here's the top 20 sequences. So initially, we have 2 primers and a 40-mer in the middle. Single-stranded, so 2 primers, 40 bases, all the random choices. So all those types of 40 mers plus the 2 primers are there. But they're very rare, so you never see them individually. But as time progresses, some bind to that target better than others. So initially, their frequency is pretty small. You can't see them. I mean, only you can see them if you sequence everything, but you wouldn't be able to show it on a graph. But after around between 4 and 7 passages, you get this big, some of the winners really win. So the top 20 winners, so here's replicate 1, they're 50% of the population. 50% of the population is made up of the top 20 individuals when you do selection on beads. What if you do selection on cells? So this is the part of the experiment where everybody says, don't do that, that's stupid. Sorry, I shouldn't say. Don't do that, that's not, what's your hypothesis, blah, blah, blah. So our hypothesis was that cells are complicated, and there's lots of opportunities for different kinds of DNA to bind to them. So you're changing the selective regime. And so the utility here is that you can see the consequences of a simple ecology, which is a bead, to a complex ecology on the subsequent evolution. So you're leveraging this simple versus complicated, which is a classic thing in ecology, and you're leveraging the comparative method, right? So I'm making a comparison. I'm not just doing one thing. Here, because you have this comparison, I can compare them together, right? And so in this system, notice the number here is 500, not 5 times 10 to the 4th. So there are no winners here. There are things that lose. But there's not a lot of winning. And there's something I haven't told you. And what I haven't told you is that when you do it on beads, most of you are getting a screen. Most of it is a screen. So 76 to 80, that's the ancestral length. There's a little bit of variation here because whenever you do make a library, there's some errors that creep in. But after 8 rounds of selection, they're mostly still 76 to 80 mers long. But when you do it on beads, the vast majority of them are way bigger. This should say 81, but whatever. They've grown a lot. How is it they're doing that? Well, so here's the thing I probably should have shown first. Here's 1 primer, here's this other primer, here's the 40-mer in the middle. You do 8 round selection, either attaching to beads or cells. And on beads, you have 1 primer, another primer, and the ones that bind to the beads best, because some sequences bind better than others, you get, that's what they do. And so the biggest winners win. But on cells, there's lots of different ways of binding to a cell. And so you have a tremendous amount of diversity maintained throughout the selection. And because of that, they do something else. They start getting internal repeats. They cooperatively, via sex basically, that these these single-stranded DNA start having sex, and they start making internal primers. And so they get way longer. And because they're internally priming themselves, they replicate faster. Because there's lots of different ways to bind. So there's a lot more opportunity for evolution to happen when you maintain diversity, which is an obvious thing, which everybody knows who knows anything about population genetics, that maintaining diversity accelerates adaptation. And so here, because we're building on cells, we're able to maintain a lot of diversity, and because of that, we're able to see this recombination happen. And they evolve really quickly, and you get these complicated shapes, which allows for a lot more interesting adaptation to pursue, to happen. Okay, so hopefully I illustrated for you how complexity can really easily evolve, and it can really easily evolve even in experiments. They're really simple experiments. And you can test ideas about whether cancer limits, evolution of multicellularity, whether there are common constraints for pretty strong extremophiles, right? So these are bugs that live in 250 grams of salt per liter, so it's pretty salty. Not as nasty as the things that animals show, but still pretty tough conditions. And you can even see that complexity rapidly evolves in a system that doesn't have there's no catalytic ability. The only thing we're asking them to do is bind. And we leave open opportunities for adaptation to occur. And by leaving open opportunities for adaptation, instead of closing them all down, we see adaptation proceeding. Okay, so these projects involve a lot of people, some of whom are, all of them are, well, hopefully all of them are here. They should all be here. So Bea and Nally have already left, but Pooh is here, so you can ask her some questions about the multicellularity stuff. So we have the multicellularity team, the archaea eukaryogenesis team, and Steph is right there, and then we have the origin of life. So these are like the biggest questions we can ask in biology, and we're trying to answer. We're trying to get them. We're trying to get some insight into them. So the things that I've talked about thus far are like big major biological transitions: the evolution of multicellularity, the evolution of eukaryotes, the origin of life itself. These are evolutionary transitions that change the world. And so here they are. And these are typically called, let's see if I can remember which button to push, major evolutionary transitions. And what they evolve is additional complexity in genetic architecture. So multicellularity, you have multiple cells. In eukaryogenesis, you have multiple genomes, basically. And the origin of life, you have, well, the sort of beginning of a kind of coherent genetic architecture. Right? These are called major evolutionary transitions. And these are the ones that we've been investigating with experimental evolution now for, 15 years. But it turns out, so whenever I talk about them like that, the next thing that happens after they say why multicellularity takes so long is they then say, well, what about photosynthesis, nitrogen fixation, methanogenesis? They weren't important? And I said, well, yeah, they were important. Well, weren't they major evolutionary transitions, Mike? I said, yes, they were, but what are we supposed to do, right? So Valeria and Luis came up to Minnesota earlier this spring, and one of our colleagues, Jim Elser, who's done a lot of work here, was there as well, and he gave us talk about the importance of phosphorus, which is obviously connected to Glossinianacus because there's hardly any free phosphorus. And Jim's always talking about the wonders of phosphorus. And I'm always like, okay, Jim, sure.
That's true.
And yeah, absolutely. He's constantly on the wonders of phosphorus. And I was like, but phosphorus couldn't have been limiting. Could it have always been limiting? Because why would organisms have so much phosphorus if it was always limiting? That doesn't make any ecological sense. So what we dreamed up is that, in fact, there are major eco-evolutionary transitions. That, in fact, that would capture a lot of what other people think is important in the history of life, that transform the ecology. And what these are about building new niches in ecological interactions. So Valeria introduced this idea in her talk. you probably don't remember, it's so long ago now in the meeting. But unfortunately, you can't read this. This is Resource Competition and Community Structure by David Tone. And so thank you. So he's what you know from ecology is that resource limitation and energetic limitation is super important in structuring ecological communities. We know this. That's quite a lot of ecosystem ecology. That's the default for ecosystem ecology is resource and energy limitations. That's how they structure it. And so why isn't that important in the evolution of those ecological communities? And of course the answer is, of course it's important. And so, how do we know it's important? Well, this isn't the, so Valeria showed this one that Luis's hands up and you can sort of see those bands, but I couldn't see those bands. Maybe you probably can't see them where you are either. So here's the bands of here's microbial bands. And what you'll see, if you could see it, is that in fact, what you'll see is a whole bunch of layers that happen one after the other. And what these layers refer to is eco-evolutionary transitions in the history of the sort of early life, right after the evolution of cellular life. So the first thing, so right at the beginning you get methanogenesis and acetogenesis, right? So you get carbon fixation and energy. You get anoxygenic photosynthesis and aerobic heterotrophs. These evolve following those. Oxygenic photosynthesis and aerobic heterotrophs. So all of these are solving problems with carbon limitation, nitrogen fixation, and energy, availability of energy. And they occurred sequentially. In fact, that's already known. That's already known. And the only thing that hasn't been put together is that in fact it happened sequentially, and the consequences of that, the reason it happened sequentially is because of classic resource limitations in ecological thinking. So what we're arguing is that you can see these eco-evolutionary transitions because they change the ecology, and they are an evolutionary transition. I have spam apparently, and you can see them, interestingly enough, in the microbial layering, because this microbial layering is actually conserved through the history of life, which is a nice example. So the kinds of approaches that we do, so we don't just do experimental evolution. We also think about how we can connect to the field biologist, of which I'm not really one, to get insights from them. So our team, so Bea and now we're here, so we have a team of folks who study microbial ecology, and eco-evolutionary origins. Pooh is going to talk about predator-prey, and now he studies archaea and bioinformatics. So a really collaborative team. We don't all work on all of each other's projects, but we like to talk about ideas. So I encourage you to talk with us here today and maybe consider moving as far north almost as you can. Consider in the ice cold north of the United States and join us at some point, hopefully, with us there, because we really appreciate talking with you. And thank you.
Thank you. Thank you very much. Mike, I've worked with multicellular cyanobacteria, those anabaline linear ones, beautiful ones, and reading about the multicellularity of these organisms, I've always read that multicellularity evolved independently a number of times, and they even say numbers like 16, 17 times. Can you clarify that point?
So I think I cut that slide out. So among eukaryotes, it's probably at least 25 times that it's independently evolved. And probably the reason for that is the background ecology of those is facilitating the independent origin, right? So it turns out for plants, you get many of those, the ones that are capable of fixing, do photosynthesis. Those organisms provide independent origins for multicellularity. They're quite different than metazoans, right, and fungi. So the environment they're in is a kind of filter for the kind of multicellularity, the reason why you get an independent origin. And the ones that for the multiple plant-like ones, of which there are actually a number of those, they're all green, they all kind of look like plants, but they're not, they're independent origins. That's, I think, an interesting question, and I don't think we know the answer for that. Now for cyano, I mean, they're clearly multicellular, but they seem to be stuck, and no one really knows why. Yeah.
Thank you, Mike, for a very interesting talk. Let me see if I can make up my mind. So these microbial maps, do they have always the same structure? They have a conserved structure? Have somebody made some comparative biology? Or?
So I wouldn't say they always have the same structure, because clearly some of them don't. But it seems like they have, if you look where they have most of the bands, they seem to form in this particular architecture that a top and a bottom, right? And the top and the bottom goes this way. So some places don't seem to have all of them. I mean, I think they don't generally miss the middle, but you might not have the bottom. I think that's what you see. And so my colleagues, I wouldn't be right to say, Larry, you can answer this question, but maybe I could say that. So that's what people have told me.
Hello, thank you so much for the talk. Evolutionary transition, I think that is a very nice concept, but I didn't catch it completely.
Yeah,
I think it's quite complex, so I would like to know what it means. I understood that this is like a new concept related with the what drive the complete evolution, if it's in that way, how this will happen. So because I thought that it was a distribution, the stratification based on the metabolic necessities, but it is a way of transition evolutionary. I got very interested.
All right, so if you're carbon limited, right? So initially we have all these things that possibly heterotrophs initially near the origin of life, but carbon is possibly limiting because basically most of the carbon that you have available isn't really available. It's not organic. So evolving a methanotroph and a cetotroph releases that carbon limitation, right? But that means that you have a new limitation arises. So that's basically it. So you release one resource limitation, and that opens up a new limitation, right? And so that proceeds until it basically becomes phosphorus limited. And phosphorus limitation is very difficult to relieve because phosphorus takes itself out of the system. And that's basically the idea, at least with respect to microbes at the base of life.
Hello, thanks for the talk. And I have a question. What do you think about the role of the eco-evolutionary aspects about the last universal common ancestor? I ask this because in the last inference proposed that LUCA maybe can be a cetogenic organism that produce acetate and can be the first carbon source for another microorganism.
Right. I think that in the last 5 years, there's been a lot of advances in our understanding of those transitions and the possibilities. I think people are really thinking hard about the ecology of those systems and not just the genetics, which is what you kind of referred to. I guess my thoughts are that I think there's probably more things we don't know than we still do know. That's probably what I would say. So if you said that 10 years ago, everybody would be like, well, we don't know anything. And between 5 years ago and now, we actually made a lot of advances, particularly with comparative genomics, really. It's made tremendous advances. But it seems like there's still, which archaea is it? Oh, it's this archaea. No, it's this archaea. I mean, whenever I hear that, I'm thinking, we don't know enough. I don't want to put all my eggs in this. Oh, it's that archaea, and then 5 years later, it turns out it's this archaea. That tells me we don't know enough. But I think that having these hypotheses actually is really great.